Hello, everybody. This is Mary Annarella coming at you live for edition 2 of I Can't Believe We Had Another Snow Day on a Tuesday and Thursday. So what I'll be doing here is recording a lecture that we missed right before the exam. So I'm just catching you guys up. This is Bio229, Nestor Microbiology, Control of Microbial Growth in our book by Nestor. So what I'm going to do here, and sorry, apologies for, not going full screen on you, but what I want to do is, because I don't have a board to write on for you all, I am going to toggle back and forth between this Word document and the PowerPoint so we can follow along. So the Word document will be basically what I would be writing on the board as best I can type. Just a heads up that my M key, my letter M key, is sticking, so you will probably notice that I have problems with words that have the letter M in it. There may be an expletive on occasion. I cannot guarantee that there won't be any. I'm a little giddy on a Saturday evening recording this for you all. But anyhow, control of microbial growth. Oh, look, the M key worked that time. Anyway, we're into chapter 5. This will definitely be shorter than our full lecture time because you guys aren't here to ask questions and you can't see me when I'm waving my arms around to try and demonstrate some sort of concept. So it'll go probably about 45 minutes or so, but I'll get through all the material for you all. Unfortunately, I will not be able to answer any particular questions in real time for you. So if you have any questions about this material, please ask in class when we meet again, and I would be happy to clarify for you. Anyhow, let's get on with chapter 5, control of microbial growth. Oh, and again, I'm not going full screen on you here because then I can't do control tab in between the Word document and this. I'd have to escape full screen capture. So anyhow, escape the full screen for the slide. So we're just going to leave it this way. So apologies if you don't prefer this. All right, so types of control. First, let's talk about what is the purpose of what we are trying to do here if we are trying to get rid of some microbes. Sometimes we just want to stop them from growing. Sometimes we want to kill them. And sometimes we don't want to kill them. We just want them to live someplace other than where we don't want them to be. We just want to remove them. A nice example of removing bacteria would be washing our hands. Regular soap does not destroy bacteria, it just removes it from our hands, and we hold our hands underneath the water faucet and we wave bye-bye to them as they go down the drain. Anyhow, there are both physical and chemical methods to do this, and we will talk about both of them. All right, but first, a bit of history. So some names you guys need to be aware of. Louis Pasteur. Remember, he is the swan-necked flask dude who demonstrated that microbes exist in the air and that his broths would stay sterilized until they were exposed to only the air. Prior to that, people did not realize that there were particles and living organisms that you could not see with your own eyes that existed in the air. He was the first to propose that microorganisms actually cause disease. This concept is actually called germ theory, and that is something that I would write on the board. Pasteur. Oh, can I spell in French? Can I type in French? We'll see. Pasteur. So his work contributed to germ theory. The idea that a particular microbe can cause a particular disease. His work definitely supported that theory. Semmelweis. This is a very interesting person. Oh gosh, I forget what his first name was. Was it Anton? I'm not sure, but he was a Hungarian citizen who was working in Vienna in the mid-1800s, so around 1850. His work was actually before Pasteur and Lister, and he was the first to suggest that doctors wash their hands. This is because, and this is a little story here, This is because he was a physician who worked at 2 clinics. And at clinic number 1, and here's where on the board I would draw a little picture of a house of a clinic. At clinic number 1, there was 10% mortality rate in the maternity ward. So what this meant is that 10% of the women who came there to the clinic 1 to give birth, 10% of them died. Let's put this down. Semmelweis. Hand washing guy. Clinic 1 had a 10% maternal, see, there's my M key sticking again, maternal. Really got to take this laptop in or just buy some canned air and blow it through the keyboard. But there's some piece of crud that's blocking my M key. Okay, clinic 1 had a 10% maternal mortality rate. Let's tab this over here. Whereas clinic 2, Semmelweis worked at 2 clinics, clinic 2 had a 1% maternal mortality rate. Rate in, oops, how did I get an R up here? Oops, rate in the maternity ward. Okay, when a woman gives birth, there is a lot of blood, okay? And so a common member of the normal flora, of the normal vaginal flora, is strep bacteria, Streptococcus pyogenes. But there's so much blood that if a woman gets an infection with that, she can die from that. And that was much more common at clinic number 1, at clinic number 2. Okay, the deal with clinic, so Semmelweis is like, what the heck is going on here? I mean, maybe 10% doesn't sound like too high of a death rate to you, but wow, that's huge. Are you kidding me? 10 women going to give birth, and one of them isn't going to make it? I mean, that's terrible. So Semmelweis was trying to figure out, well, what is the difference between what's going on at clinic 1 that had a higher maternal mortality rate than what was going on at clinic 2? What was clinic 2 doing right that clinic 1 wasn't doing right? They appeared at face value to be doing the same darn thing. After testing out several different parameters, he finally concluded, you know what? I have noticed that at clinic 1, that the medical interns were visiting the morgue and would leave the morgue to go directly to the maternity ward. So at clinic 1, physicians, went to the maternity ward to help deliver children. There's my M key. Right after visiting the morgue. Did I spell morgue right? Yes, I did spell morgue right. You guys know what a morgue is? A morgue is where the dead bodies are stored, and not stored, just stored for a short period of time, where they are examined to determine the cause of death or perhaps to be identified by the family members. So residents were going and doing autopsies on dead bodies, which many of which may have died from bacterial infections, and going directly to the maternity ward to help deliver children. So Semmelweis suggested that after leaving the morgue, residents, all individuals, wash their hands. So Semmelweis recommended that everyone wash their hands after leaving the morgue. And again, there's my M key. You're going to hear all about this M key. Anyhow, this seems to make sense. Well, this totally makes sense to us today. I mean, you use the bathroom, you wash your hands. You touch something grody, you wash your hands and you wash them well. But back in the mid 1800s, this was crazy talk. Semmelweis was made fun of. He was ridiculed by his colleagues. He eventually lost his job over this, and died shortly thereafter. I don't know, died of a broken heart. But after his death, residents and other physicians began actually washing their hands, and it reduced the death rate at this clinic tremendously. So we have Semmelweis to thank for just learning about and determining that we needed just a general standard of cleanliness, and that includes washing your hands. Everybody in class here did the hand washing experiment, and you saw what was on your hands just for transient bacteria. Could you imagine if we had all worked in a morgue without gloves on our hands, and then went and delivered a baby? Yeah, bad news. I mean, so when you even think of ER, private practice, and some other doctor and physician TV shows, oftentimes you see people scrubbing up before surgery. It started with this guy. And even though I'm at home, I still have not turned off my phone, and you can still hear me getting texts and notifications on this. Oh, wow. Oh, unless you guys didn't hear it, my phone just went ba-bonk for a notification. Anyhow, not that you guys care about that. But, oh, wow. Why won't this work? There we go. All right, Semmelweis. Oh, Lister. What does that sound like? Lister, like Listerine. Lister came a little bit after Semmelweis, and he began using antiseptics during surgery. Wow. Can you imagine not using an antiseptic during surgery? His was the first idea to do this. He was the first to start doing this. Okay, Koch, or perhaps the guy's name is pronounced Koch. It's probably pronounced Koch. Okay, this guy was famous, incredibly famous microbiologist. He identified and isolated microbial pathogens. More than that, he was able to link particular diseases as being caused by a particular microbe. So, for example, Bacillus anthracis is what causes the disease anthrax. Koch is the first person to actually make that link. So not only that, and also determined that tuberculosis was caused by a Mycobacterium tuberculosis. So Koch is a very famous person. So he linked diseases to particular microbes, and he won the Nobel Prize for that. Nobel Prize in Medicine for it. Also, he was the first to use agar as a solid growth medium. Okay, so all the agar plates, he was the first to use agar in there. Before using that, he actually used to grow, instead, in order to do a streak plate, before he started using agar plates, boring agar plates, he used slices of potatoes. Could you imagine doing a streak plate on a large potato slice? You ever know? Do you know what a potato slice probably looks like after incubating it overnight? It's going to be all brown and grody looking. Okay, and his wife was like, why are you using those brown and grody looking potato slices to grow bacteria? And he's like, well, because I need some sort of solid medium, and the potato slices have all the starch in there, so they feed my bacteria. She's like, well, why don't you grind up some starch and put it into this agar stuff? His wife was making jam preserves, and as a thickener, she was using agar. Agar, again, as you know, is a plant-derived product that is similar to gelatin, but it's harder and sturdier than gelatin. So he's like, oh, okay, I'll try it. And he's like, wow, you are a genius. So we can credit Koch's Koch's wife for all the agar plates that we use, because it was her idea. All right, so a bit of microbiological history. Oh, and Koch. Koch also came up with a series of criteria that need to be fulfilled in order to link one disease to a particular microbe. And those are actually called Koch's or Koch's postulates. Postulates. These are criteria that must be fulfilled to link a particular disease to a particular microbe. That was Koch, and we will study his postulates at another time. Okay, where are we going with this? Ah, yes, some terminology. So just to make sure we know what we're talking about, when we say sterilization, we talk in terms of destruction of all life forms sterilization, like for surgery, for example. But there is a term, commercial sterilization, that is used by the food industry. And what it means is that there are fewer bacteria in a particular food sample, fewer numbers of bacterial cells in a particular food sample than will cause human disease. So that is commercial sterilization. So it's used by the food industry. And it refers to keeping things under the number of bacterial cells that can cause disease. Commercial sterilization. So like in canned goods and the like. Okay, disinfection. When we disinfect something, we talk about disinfecting things on non-living surfaces. So let's say for non-living surfaces. Whereas if we're talking about using, say, a chemical on living tissue, we would call that an antiseptic. So you would not use a disinfectant, say, on your skin. You would use a disinfectant on your kitchen countertop. You would use an antiseptic on your skin, for example. You would not use an antiseptic on your kitchen countertop or your kitchen sink or in your toilet. You would disinfect your toilet, you would use an antiseptic on your skin. So there's a difference between those two. Okay, if we say that something, okay, side means killing and static means to prevent growth. So what we mean by that is if something is bactericidal, means it kills bacteria. If something is bacteriostatic, it merely stops the growth of bacteria. So something like bleach will kill bacteria. Bleach is bactericidal. Whereas certain things like certain mouthwashes merely stop the growth of bacteria. Those would be bacteriostatic. And then pasteurization. Pasteurization does not sterilize. Pasteurization is not sterilization. That is so important. Let me repeat that again. Pasteurization is not sterilization. What is pasteurization, then, you asked? Pasteurisation, to keep it French. Pasteurization merely is heat treating a substance, usually a food substance, so that you kill off most of the pathogens. So you're killing most of the bacteria, but not all of it. So using heat to destroy most, but not all, microbes. Let's say microbes, because we could be talking fungus too. Okay, so this is for things like milk. And there's my MK again, sticking. Milk, apple cider. Someone in my afternoon class said that they were reading something about almonds being pasteurized, which seems kind of odd to me there, that you would sterilize, that you would use pasteurization to sterilize almonds, because pasteurization usually uses moist heat And moist heat would take the skins off of the almonds, so those would technically be blanched almonds. But there was the article. So somebody's pasteurizing almonds. But usually pasteurization is used for milk and apple cider. So we heat usually to 72 degrees C. Usually it's fairly short, just for like 30 seconds or so. Although you can pasteurize at a higher temp for a shorter period of time. That would be high temperature, short time pasteurization. A lot of juices for juice boxes are pasteurized as well. But you guys know that if you buy a gallon of milk and you let it sit in your fridge, even if you don't open it, if you let it sit in there for 3 weeks and you open it up, you know that's going to smell, because there are still some microbes in there. It's just destroyed most of the pathogens, and it allows for a longer shelf life. So it's usually used for liquids. Apple cider is another thing. It's really difficult to find unpasteurized apple cider anymore. Mostly because, okay, you guys know that apple cider is basically pressed apples. But if you think about, a lot of apple cider is made from apples that have dropped to the ground. Those are called drops. You can wash them off, but think about what goes through an apple orchard. There are squirrels and possums and deer and cats, dogs going through apple orchards, and they're pooping there, and the apples fall to the ground and they touch the poop, and you guys know that there's bacteria in poop no matter what animal it came from. So usually a lot of places will wash their apples, but as an extra precaution, we pasteurize it to kill all of the coliform bacteria that could make us sick. So it's difficult to find apple cider that has not been pasteurized. I wouldn't drink unpasteurized apple cider unless it was from a cider mill that I really trusted, where I knew they really washed their apples really well, or that they did not use apples that had dropped to the ground, that they used only apples that they picked off the trees. But even then, those probably should be washed, because birds are in the trees and birds poop, and I don't really want to drink apple cider that has bird poop in it. But that's more information than you guys probably need. Take home message: pasteurization is not sterilization. FYI, this is a Saturday evening, and I am not used to doing a lecture on Saturday evening, so you get the little extra jokes, the little extra TMI about my not wanting to drink apple cider that has not been pasteurized, just for free. Okay. All right. So how you want to sterilize, or how you want to pasteurize, or how you want to treat a substance depends on, well, what type of microorganism you want to kill off, what the setting is. So for the setting, for example, things in, for example, a room where surgery is being done need to be sterile to a higher level than, say, your kitchen or bathroom. What type of microbes are you trying to kill? Are you trying to kill fungi? Are you trying to kill coliform bacteria? And what are the instruments that you're trying to sterilize made of? Are they made of plastic? Are they made of metal? Are they made of glass? If things are made of glass, well, then you could bake them in the oven. You could put them in an autoclave. If things are made of plastic, you're not going to want to bake them in the oven, because plastic's going to melt in the oven. Ask me how I found that out when I worked in a laboratory years ago. Anyhow, you need to take into consideration what things are made out of and the level of cleanliness that you need. All right. Heat. Okay, so physical ways that we kill bacteria, okay? Heat is a physical way that we can kill microbes. There is dry heat versus moist heat. Dry heat, baking stuff in an oven. Moist heat would include boiling things, or pasteurization, or in an autoclave. But both are physical processes, and moist is definitely better than dry. And we'll talk about why in just a moment. Oops, I cannot be clicked on a text box and forward through here. Here we go. Oops. Oh, no. Okay, so yeah, we got to talk about dry heat. Dry heat would refer to an oven. Heat in an oven. Okay, it is not as effective as moist heat, but it does work for things such as destroying certain proteins. So not as effective as moist heat, but can destroy certain proteins. So for example, there's a protein called RNase is destroyed best with dry heat. RNase is an enzyme, it ends in ase, that breaks down RNA. So if you're trying to isolate RNA, you want to make sure that none of your instruments have RNase on them. And it turns out that RNase is all over your fingers, so you can't touch that stuff. So we bake a lot of things that need to be used for isolating RNA. So we got that. But baking does work. Moist heat, boiling, pasteurization. Missed the S in pasteurization. That's going to bug me. And also autoclaving. Autoclaving is high temperature and pressure. Okay, so like you can boil something, but you know most things are water-based that we're trying to sterilize, and so the temperature at which water boils is 100 degrees C, but you need to get higher than that temperature to destroy certain microbes. So how are you going to get higher than 100 degrees C? Autoclaving is how you get higher, how you get hotter than boiling. And that is because the autoclave, and you've probably heard this thing beep in the laboratory. When you hear a distant beeping sound, sometimes it usually means that the autoclave is done. An autoclave basically is a vacuum with water jets around it, and we boil the water that's in there to steam, but then we pull a vacuum on it. So the steam, the air and the water that's in there wants to expand, but if you don't let it expand because there's a vacuum on there, that drives the temperature up like whoa. I forget which chemical law that is. Maybe it's Boyle's law, but an increase in temperature, an increase in pressure causes an increase in temperature. So we increase the pressure, and that drives up the temperature to about 120 degrees C, which is really freaking hot. Okay, so about 20 minutes in an autoclave will sterilize well. But to kill prion proteins, I can't talk and type at the same time. You need up to 4 hours of autoclaving. So that's a lot. But just about everything we work with in the laboratory has been autoclaved. So it's a very useful thing. Oh, and your dentist's office uses an autoclave, and they have just a very little small benchtop version of an autoclave, and they autoclave all the dental instruments. You know, after they clean them, they autoclave them because, you know, they use those dental instruments in a lot of different mouths, and it's not all disposable. So autoclaving takes care of that. All right, another physical method, filtration, does not kill microbes, but it removes microbes, and we talked about this a lot. You pour your sample, your liquid sample, into a funnel over which there is a membrane, and the membrane filter has little holes that are small enough to let the water through, but actually traps the bacteria on the filter. You throw the filter away, and all of the water that comes back down, that comes through the filter, is basically sterile. So these are used for water for purification systems. A lot of people who go backpacking or hike the Appalachian Trail are going to be backpacking in places where there is not a secure water, not a reliable water supply, can actually filter their water that way. And these air filters are also used in air purifiers. You guys have probably seen those, like in certain hospital settings. Sometimes people have them in their homes. Oh, and also there are some solutions that we use in the laboratory that if we autoclave them, they just get to be thick and unusable. So for example, there are like soap solutions that we need to use for certain procedures in the laboratory for isolating, like plasmid DNA out of bacteria. And you want to use a little bit of basically an SDS solution. Well, SDS is one of the primary ingredients in a lot of shampoos. And if we autoclave it, it basically turns into a thick, creamy looking shampoo, which might work great for a shampoo, but it doesn't work so well for pipetting. So instead of autoclaving that SDS solution, we actually filter it. I'm not going to ask you about SDS and filtering it on the exam, but that is just to give you a real life example. All right, another physical way to destroy microbes is through radiation. Gamma rays, X-rays, and UV light will all damage DNA, and we destroy bacterial cells by damaging their DNA with gamma rays, X-rays, and UV damage. Now, X-rays, a lot of us here have probably had an X-ray at some point. If not on most of our body, we've probably had dental X-rays. And you know how they usually put a lead apron over you to cover the rest of you, and you're like, oh my gosh, what about my head? But this X-ray energy is focused right on your teeth, and it is at a low enough dose that it is fine for your cells. But if we use X-rays at a higher dose, we can destroy bacterial cells. But UV light will also damage DNA, and it will damage DNA in your cells, which is why we tell everybody to wear your sunscreen, because it can damage your DNA. We actually use UV irradiation in certain laboratories, in certain flow hoods, where we're going to be doing tissue culture, or culturing cells that really need a super, super duper clean air environment. And overnight, we will turn on UV lights, like sometimes in the entire laboratory, but definitely inside the hood, to destroy bacterial cells by damaging their DNA. Now, microwave energy. Microwaves do not damage DNA. Let me repeat, or let me write this down. Microwaves do not damage DNA. Microwaves destroy microbes by generating heat, and heat kills them. So you can use a microwave to pasteurize, for example, but you would have to microwave the daylights out of something to sterilize it. And why would you want to use that? Why would you want to like sterilize your food? I mean, if you microwaved it to death, the food's going to taste horrible. There are much better ways to make your food palatable and edible. So we generally do not use microwaves to sterilize. Pasteurization, somewhat, sure, yeah, but and the other thing I wanted to add to you, talk about, a lot of food is irradiated. So for example, and not like packaged foods, onions and potatoes and things like that are stored in trucks, that are stored in silos, and stored on farms before they are picked up into trucks and held at warehouses and then trucked to Stop and Shop. A lot of food is irradiated, not to kill microbes usually, but for pest control, to destroy mice, to destroy mosquitoes and things like that get in there. I mean, seriously, otherwise you'd end up with rat poop and the like, on your onions. So we actually irradiate food more for pest control than for killing microbes. So irradiated foods, we irradiate these mostly for pest control. And Oh, and there's my M key. You are just not going to hear the end of this M key. We irradiate these mostly for pest control, and I looked it up going like, well, how do we know what food has been irradiated and what has not been irradiated? It's supposed to have a little label on it somewhere. I mean, either on the little sticker that would be like on the onion or somewhere on the information on the little sign saying onions, blah, 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 at the grocery store. A common misconception about irradiated food is that it is radioactive. It is not. The gamma rays and the X-rays that are used are not used in the presence of humans. You have to leave the room before you press the button when you do an X-ray, for example, and it's the same thing when you irradiate food. But that irradiation does not stay in the food. It is not radioactive. So radioactivity is something other than using gamma rays or X-rays. That's like saying that an X-ray room, and I just found a typo up here. That's like saying that a room where X-rays are taken, that room is radioactive. It's not. It's not. So it's very safe. Although irradiating food can alter the form of some of the vitamins to make them less absorbable, but that's true for any type of food preparation. When you heat a food, you change it. When you heat it by boiling, you change the composition of it. You change the shape of the proteins and some of the vitamins too. Sometimes it's for the better, sometimes not. So, whereas you wouldn't want to eat an entire diet of irradiated foods because you would be missing out on some vitamins, that's true for you would not want to eat an entire diet of exclusively boiled foods. You want to vary your diet. So there's your public service announcement right there. Irradiation, other means of physical methods of control. Low temperatures do not sterilize. It merely slows the growth of mesophiles, and we actually use low temperatures to store bacteria in laboratories. We also use desiccation, and lyophilization, which is basically freeze drying. It stops the growth, but it doesn't destroy. It doesn't sterilize food. So if you buy freeze-dried strawberries, if you open those up to the air, if you rehydrate them even with sterile water, there's going to be some bacteria on those freeze-dried strawberries that will eventually grow if you let them sit around for, long enough. But you don't generally do that. You freeze-dry your food, you rehydrate it, you eat it. We actually use freeze-drying, lyophilization, to store bacteria in the labs. In fact, when we purchase some bacteria for the laboratory, we usually buy it all dried down like that, and we just squirt some water onto it and give it some media, and it starts growing like crazy. All right, water availability. If you add a lot of salt or even sugars, high enough levels of sugars will inhibit bacterial growth. We talked about osmotolerant and halophile bacteria that don't mind having a lot of salt in their environment, so it would not slow the growth of those, but we do use salt and high sugars to prevent the growth of microbes. We use that for food preservation. So, like bacala, we talked about salt cod, salted foods, is a means of preserving those foods. High sugar content will do that as well. Jams and jellies are an example of that. So we boil it down. We actually can them, but we don't generally further process those. We don't have to put them into an autoclave. The high sugar content actually inhibits the growth of a lot of bacteria. So those are physical methods. We have chemical means of controls. So when we're talking about chemical means of control, we're talking about antiseptics and disinfectants. Disinfectants are used on non-living surfaces. Antiseptics are used on the body. If we're talking inside the body, not on the body, but usually inside the body, we are talking about antibiotics. And again, I can't forward unless I'm not clicked on a text box. So here we go. So chemical means of control. All right, we use alcohols. Hand sanitizers. Almost all hand sanitizers are alcohol-based. So chemical. So alcohols. Alcohols. Did I spell that right? That just does not look right, but because that H doesn't belong there. There we go. Alcohols. All right. 60 to 90% alcohol will destroy bacteria. most bacteria. I think you need at least 80% to kill viruses. So your hand sanitizers, your Purell. If you're in my lab, we used Purell, and I believe that is actually a 75% alcohol solution for the one that we bought. But that will kill bacteria. The only problem with hand sanitizers is that they're very drying. Alcohol is drying on your skin. And so this time of year, especially in the winter when the air is very dry already, you got to be careful not to use that too much. So again, that's not something that you use in place of washing your hands. That's something that you use when you can't wash your hands. If you pull over and use a bathroom at a truck stop and that bathroom is really gross, you might want to use hand sanitizer. Or if you're in a spot where you have to touch something dirty and you need to wash your hands, but there's no sink around, you can use a hand sanitizer. So that is alcohol. What else do we have? Aldehydes, formaldehyde. Formaldehyde. It's used in embalming. The embalming process, preserving a dead body. It destroys microbes that would basically compost the body, that would degrade the body. So that's what we use for formaldehyde. It is toxic stuff to you. I'm going to be careful using that. It is really super smelly. We're going to nix biguanides. You can nix that for sure. Okay, but halogens, chlorine and iodine, that's what the Cl and the I stand for. So let's see, halogens. Use chlorine in pools. And hot tubs to kill bacteria. We use iodine. You guys know what we use iodine for. We use it on skin. We use iodine solutions on skin for medical procedures. So for surgery, I spelled surgery, blood donations, vaccines, anytime we need to have a blood test, anytime an IV needs to go in, they usually use iodine on the skin to destroy bacteria. Metal compounds right here. This is one of my favorite. Ag, that stands for silver. AgNO. Do I have a little subscript here? Yes, I do. Silver nitrate drops are placed into the eyes of infants, but because it destroys Neisseria gonorrhea and the microbe that causes chlamydia. So in the eyes of newborn babies, the silver nitrate destroys Neisseria. Can I spell gonorrhea? G-O-N-O. I think it's 2 R's. H. Gonorrhea and chlamydia. Trichomonas. Let's just go with chlamydia. These are both STDs. STDs cause microbes that cause STDs. Sexually transmitted diseases, in case you didn't know what STD stood for. Sexually transmitted diseases. It is so much easier, and they're cheap too. It is so much easier to just put these drops in the eyes of every newborn than to specifically test every single pregnant woman for gonorrhea and chlamydia. No, I spelled 2. I mean, yes, you can ask, but not every woman has prenatal care available to her. And so any child that is born in a medical facility would automatically have silver nitrate drops placed in their eyes. Okay, phenolic compounds. This involves phenol. Phenol is this orangey stuff, and we use it in the laboratory on occasion. It's similar. It smells similar to formaldehyde. It looks kind of similar to formaldehyde, but it it has some antimicrobial properties, and that won't harm your skin as much as formaldehyde would. But phenol. They put it in several topicals, like camphorphenique has a phenol-like compound in it. And that's really all I need to say about that. All right, so my spiel. Antibiotics are no substitute for prevention and control. You guys know that antibiotic resistance is a huge problem in the medical community, and doctors are very wary to prescribe an antibiotic for someone that they aren't absolutely sure, really sure, that where they actually have a bacterial infection. So I just wanted to briefly talk about antibacterial soaps, because antibiotic resistance is caused in part by overexposure to substances that microbes can internalize. One substance that has been everywhere is something called triclosan. It's also called Microban. I haven't heard ergosan in a long time. But there was evidence that triclosan, which was in everything, I mean, 5 years ago, you could not go to Bed Bath & Beyond and find a liquid soap there that didn't have triclosan in it. They were all antimicrobial soaps, antibacterial soaps. They were putting this stuff in everything. Studies were showing that the stuff was actually capable of being internalized by bacterial cells, which could lead to resistance, and then you can't use it anymore. When a microbe is resistant to something, you're screwed. You can't use it even when you really need it. And it was just so ubiquitous. It was everywhere that finally studies were done which showed that washing your hands with plain soap was 100% as effective as washing your hands with soap that contained triclosan. And think about what happens when you wash your hands. You get some friction going with the soap. The soap forms an emulsion around the bacterial cells and the viral particles on your hands. You loosen them up. You hold your hands underneath the water, and the water washes the soap with the microbes down the drain. The triclosan wasn't doing anything extra. So 2 years ago in 3 years ago in 2016. We're in 2019 now. In 2016, the FDA said, guess what, Johnson & Johnson and all your other companies, you need to take triclosan out of hand soaps. No more. For some bizarre reason, maybe it's not totally bizarre, but they allowed it to stay in toothpaste. There is one brand of toothpaste that is out there. You can research it yourself if you like, that still contains triclosan in it. But this stuff also has been shown to be toxic to fish, to form a dioxin-like molecule when exposed to sunlight. And I don't know. It's up to you to decide whether or not you want that in your mouth. But as of 2016, triclosan is no longer allowed in hand soaps. And I know we actually had some of that stuff out for our hand washing experiment in lab, and that's because it's a really old bottle of stuff. I actually talked with Kim about it. I said, did you know this stuff has been banned by the FDA now? She's like, no, I did not know that. So we're not using it anymore. And part of the reason that the FDA banned it is that studies repeatedly found that it is no more effective than just plain hand washing. So they got rid of it because of that, and because it's got the potential for some harm. So it's not in there anymore. But anyhow, that's my spiel. You won't find that stuff at Bed Bath & Beyond anymore, or unless it's really old stuff. Maybe you could find it on a Stop and Shop shelf or Walgreens, but that's about it. Anyhow, I hope you have enjoyed edition 2 of I Can't Believe We Had Another Snow Day on a Tuesday and Thursday, and yet we do. But I hope you have enjoyed this. I know I have, and I look forward to seeing all of you guys in class on Tuesday, hopefully snow-free. The snow is coming on Monday this weekend, so here you go. Enjoy. and take it easy, and I will figure out how on earth to stop this. I have to hit Command, Control, Escape.